It's true. Both you and I have gotten cancer in the past. So if we've all gotten it before, what's the difference between people who have gotten diagnosed with cancer versus those who have never been diagnosed but have still had it before? The answer might surprise you. Hey everybody, Organized Biology here, where we make difficult biology concepts simple. And today we're talking about that very misrepresented disease, cancer. We can define it as rapid, unregulated cell growth and division. Now, if you're not a biology nerd like me and people who follow this channel, you need to know that cells make up your body. So for example, you're made of 30 trillion different cells, including your skin cells, maybe your muscle cells, maybe your bone cells. You're made of cells, so these functional units doing stuff for your body. Now, normally functioning, your cells are very happy, very smart. They grow, they divide, and they die just like a normal human lifespan. But sometimes, in the words of Mermaid Man, they become evil. They become evil because they turn cancerous, which cancer is when these cells decide by themselves, hey, I'm gonna grow and I'm gonna divide rapidly and I'm not gonna have any regulation. I'm just gonna do it. Which this makes sense because you usually notice cancer by a tumor, a growth somewhere in your body, a mass of cells that are growing and dividing rapidly. It makes sense. So we've all had these cells before in our body, but why have we not noticed it? Well, to answer that, it takes a little bit of nuance. So I've drawn a normal human cell here, and this can be virtually any cell of your body besides red blood cells. And inside the cell, you've probably seen this little sucker before in your biology class way back when. This is called the nucleus of the cell. Think of this as the commanding center of the cell because inside of it, it's got a bunch of instructions on how the cell should build itself. So think of it as like blueprints for a house, the cell being the house, and these being the blueprints. Now, the cell is made of a variety of different molecules called proteins. And we made these proteins by basically reading the instruction manual inside the nucleus called DNA. So basically the DNA has all these letters in it that code for these specific proteins, and these proteins do everything for your cells, including keeping them upright, just like the walls and the beams in a building. They also tell them when to grow and divide, as well as a variety of other things. Now I want you to think of the cell as a house. If you've just bought a house, you may want to say fly a flag outside of your house to show you who you represent. You want to fly an American flag outside of your house or your favorite sports team, and the cell is the same way. So the cell occasionally will take one of these proteins, and we're going to call that protein the flag. And in fancy terms, it's called an antigen. And that flag will be placed in a flag holder, which is also a protein. And it's a protein called MHC1. This is vital to understanding cancer here in a second, so stay with me. And this flag and flag holder will get embedded in the outside of the cell called the membrane and basically fly its flag outside of the cell. So this is a normal protein from this human cell, and it's going to get inspected every so often, just like maybe your houses get inspected by an inspector, or maybe your crazy HOA representative, whoever it is. And that inspector is going to be this nice looking cell called a CD8 Cytotoxic T-Cell. And again, think of this as kind of the inspector guy, okay? So he's gonna come by, he's gonna look at this flag and notice if it is normal, okay? If it's a normal human protein. Because think about it, the inspector has been trained to notice abnormalities, right? The bad things of a house, the cracks, the crevices that shouldn't be there. So when he notices this cell, normal protein, this inspector basically takes a lot of its tools to test whether or not this is a normally functioning cell house, right? And nothing is going to worry him. This is a normal looking cell. Great, so we're gonna leave him alone. However, the inspector is also trained to notice abnormalities. So let's get into that next. Let's say, for example, we had some sort of damage to our DNA, where the DNA got broken and maybe it got edited around, and now we have new DNA inserted into this region. This change in the DNA sequence is called a mutation. And mutations can be caused by a variety of things, like X-rays, like asbestos, like radiation. All of these things we know cause cancer, right? And if the mutation happens in a specific DNA sequence, it could cause the cell to make, as you probably guess, abnormal proteins. So again, we screw up the instructions, we're gonna screw up the protein. So I'm gonna draw what that looks like next. So now we've got this kind of odd shaped looking cell here, and it's growing oddly, and it's looking like it's trying to divide as well. And that could be because we've made some abnormally shaped proteins here. Again, because we screwed up that DNA from that mutation. And remember, proteins tell the cell when to grow and divide. And in this case, maybe the abnormal proteins are telling the cell, grow and divide. So this cell is now considered cancerous. And by the way, these mutations happen all the time, not only from radiation and other things like that, but it could just be random. So we've all had cells like this in the past. Therefore, we've all had cancer. However, this is what our cells likely did. You see, they're still making this flag holder. You remember that guy, the MHC1? We're still making that flag holder over here. And as I said, sometimes we like to throw that flag holder with a flag on the outside of the cell. So that's exactly what the cell will do. It'll take that flag holder and attach it to one of these abnormal proteins. And as you can see now, we are expressing this abnormal protein, now called an antigen because we're holding it as a flag. We're holding that abnormal flag outside with our flag holder, MHC1. And guess what? The inspector cell is going to come along. Right? So he's going to come and inspect this cell and see if the proteins look normal like he did previously. But in this case, as you can see, he's got this little tool here that's going to notice, whoa, that is a red flag. Pun intended. This is a red flag, right? This is something bad. This is indicating that this cell is cancerous. I'm going to show what this cell will do to this cell here. Okay, so what do we see happening here? Well, we've got this cytotoxic T-cell attaching to that abnormal flag. We've got the sharp protein kind of structured here that's poking holes into the cell itself. That protein is called the CD8 protein, which is why this is called a CD8 cytotoxic T-cell. So we've recognized these abnormal, we're poking holes in the membrane, and what that will do is it'll cause this whole cell to collapse in on itself and digest itself. That means basically this whole cell will implode, kill everything inside of it, and it will cease to exist. So, where's the connection for you? You have had cancer cells, but we've noticed that it's abnormal, and these inspector cells have killed it off before it becomes cancerous. But you've probably known people who weren't as lucky, where this cell didn't get triggered to die, right? And they actually got cancer, maybe stage 1 through 4. So what's happening there? Well, you remember how mutations caused abnormal proteins, right? Well, in certain cancer types, that mutation, that change in the instruction manual, actually makes us never make this flag holder anymore. It's called a loss of function mutation, where we now no longer have the flag holder. And if we don't have the flag holder, we can't hold that flag on the outside of the cell. And if we can't hold the flag on the outside of the cell, this cytotoxic T cell will never even come and notice that this is a cancerous cell. So with no way to tell these inspectors, hey, I'm cancerous, I'm abnormal, with no way to do that, we will continue to grow and divide and grow and divide and turn into cancer. But again, what determined whether or not you have this cancer cell killed off versus a person who gets really bad cancer? Sometimes it's just random, luck of the draw.